So a sperm happens upon an egg and goes, wow, it's an egg. And he's doing sperm things, just swimming, and then he fertilizes the egg. You guessed it, guys. We're learning about how babies are made. Just joking. But if you consider stem cells babies, then you're in for a good show. The sperm and egg produces a fertilized egg, also known as a zygote, and after 4 to 7 days, this zygote develops into a blastocyst, within which there's an inner cell mass where embryonic stem cells come from. Yes, the wondrous embryonic stem cells. But you must be wondering what an embryonic stem cell is, right? Well, this embryonic stem cell found inside the blastocyst is a multipotent stem cell, which means it could differentiate into a variety of cell types. However, it's not to be confused with a totipotent stem cell, which can produce a full-fledged organism that's self-sufficient and can survive on its own. For example, a human baby. And the thing with embryonic stem cell, because it's multipotent, it could differentiate to form a whole organ. And embryonic stem cells, they need to be tended to in order to reach a target cell type or a tissue type. They need to be tended to like a baby almost. So with embryonic stem cell, they should be cultured in a petri dish in the laboratory setting. And once cultured, they have various factors in need. For example, they need some medium supplements to push them towards differentiation. To produce a cardiomyocyte, you need B27 medium supplement. And scientists insert DNA into the stem cells so that the stem cells assimilate the DNA and start transcribing the DNA to produce proteins. But also, some scientists directly insert stem cells into tissue. We have the in vivo method and in vitro method. In vivo, the stem cells are inserted into live tissue, but in vitro, the tissue is placed in a petri dish where the embryonic stem cells are too. The benefit of is that the stem cells can differentiate based on direct contact with the existing adult cells they're trying to become. In addition, they're also exposed to the microenvironment of the actual adult cell. And the benefit of this is that they are exposed to the molecules that can induce signaling pathway in order to produce proteins that are exhibited in the adult cell. For example, with cardiomyocytes, the micromolecule you would find in their microenvironment is retinoic acid. And retinoic acid serves to develop the tissue of the atriums of the heart. With the microenvironment of the adult cells and the direct contact the stem cells have, differentiation is prone to occur. Embryonic stem cells are a wonder. With the right factors, they can transform into other cell types. Their potential for the field of medicine is truly immeasurable. For example, they can be used to develop air sacs for lungs. They can be used to develop liver cells. In addition, they can be differentiated into neurons for brains. With these type of possibilities, stem cells can transform what can be done for patients. And who knows, the development of regenerative medicine may lead to the formation of full-fledged organs.